It's my great pleasure to welcome all of you to participate in this highly important debate on lecanemab and the implications for patients and for the research in Alzheimer's disease. So we are all very excited about this milestone. It's for the Institute, UK Dementia Research Institute, an important step. We are happy to share our thoughts. And I'm also very pleased to welcome an excellent panel of speakers. I'm going to hand over the whole organization to Jonathan Schott, who is not only advisor of Alzheimer Research UK, but also for the UK Dementia Research Institute. And so he's in a very good position to overview this discussion. So John, the word is yours.
Thank you very much indeed, Bart, and I hope that everyone can see my screens, and I have here my declarations and conflicts of interest. It's a great pleasure to be asked to chair this this discussion, this symposium on behalf of the Dementia Research Institute, and to see that there's such interest. We've had more than 500 people registering, and we're aware that we have a very wide range of different participants from many different countries and many different specialties. What we're going to do is to have a discussion around lecanemab. We are not going to be, of course, making any recommendations, giving any clinical advice. Whilst the session is being recorded, it's for internal purposes only, and the views of individuals give here are theirs and theirs alone, and not for sharing. The idea is to stimulate a discussion. With that in mind, there's a question and answer box. Please write your questions in there. We're going to have a lengthy discussion at the end, so please both pose your questions and vote up questions as well. So my role is to start off by giving you a very brief introduction. My apologies for those of you for whom this will be very obvious, but clearly Alzheimer's disease is a very major problem. 60 to 80% of causes of dementia. There are about 55 million people affected worldwide. And whilst the incidence is declining in Western countries, the prevalence is increasing as the population ages, and we're going to have 75 million affected in 2030 and around 150 million by 2050. There are over 600,000 cases of Alzheimer's disease in the UK, i.e. around 2/3 of the cause of dementia. And whilst there is a lot that we can do to modify the course of dementia already, the maximum that we're going to modify all-cause dementia, and most people, myself included, think this is the maximum, is around 40%. It means that we're going to need new therapies for the majority of cases of people who will develop dementia. We do, of course, have current medications, which provide modest symptomatic benefits, but we've had no new licensed treatments in the UK for more than 20 years. And until recently, we've had multiple failed trials of putative disease-modifying therapies. What's brought us together today is a discussion about lecanemab, the results of a phase 3 study, which were much heralded and published in the New England Journal of Medicine a couple of weeks ago, and have been the discussion of intense discussion both at the clinical trials and Alzheimer's meetings in many other fora since. What are we discussing? We're discussing the results of this trial called Clarity AD. These results will be discussed in a moment in more detail by my colleague, Dr. Catherine Mummery. But in brief, this is a trial of a humanized IgG1 monoclonal antibody, which is highly selective for protofibrils as opposed to soluble oligomers, and more selective for protofibrils than plaques. This was a phase 3 placebo-controlled trial of 1,795 amyloid-positive individuals with mild Alzheimer's disease. They underwent 18 months of treatment with 2 weekly infusions and multiple safety MRI scans. Again, the top-line results is that this trial reached its primary and its secondary cognitive outcomes, showing statistically improved benefits over placebo. It had additional significant effects on a number of quality of life measures. It demonstrated robust removal of amyloid from the brain. It showed effects on several biomarkers thought to reflect downstream neurodegeneration and other processes as well. There were no excess deaths attributable that were reported in the main analysis. There were, however, excess side effects in the treated population, which were mostly mild and asymptomatic and were either infusion reactions or amyloid-related imaging abnormalities, either of edema or hemorrhage, and we'll hear about more of those later. By anyone's account, this is really a turning point for the field. If one could think 10, 15 years ago that we would have the ability to detect any of the proteins that underpin Alzheimer's disease in the living brain, that we'd be able to administer drugs that could clear these from the brain and have any clinical effects, this is really a monumental achievement. However, there are a number of issues and questions, and nobody is for one moment suggesting that this is the panacea. We've variously heard it described as the chink in the armor, the beginning of the end of the beginning, and even the chief executive officer of the company involved has been clear about where the part this may play in the armamentarium against Alzheimer's disease. And there are many questions to be addressed. First of all is the mechanism of action. How is the amyloid cleared? How can we understand and reduce the risk of ARIA? How do we understand how amyloid removal has its presumed downstream effects? The extent of cognitive benefit. Is this clinically meaningful? What was the potential for unblinding in the study? What is the risks and benefits for individuals who have different comorbidities, noting that the study did allow people with a range of comorbidities to take part, and subgroups, in particular E4 homozygotes. There's unresolved questions about increased rates of brain loss in this and other studies removing amyloid, and questions about how and when and whether we've proven disease modification so far. There are major issues about practicalities, delivery and monitoring, who gets it, how and when, and of course cost benefit scenarios. Remembering, of course, that we have a patient population that is crying out for these drugs. So after the discussions that we've had and the opportunity to analyze the data and what I think everyone believes is to be a very well conducted and presented clinical trial, there's been a lot of discussion, and the idea of this discussion and symposium is really to pave the way for the discussions that we need to have in the future. And maybe is to say, well, we have an amazing opportunity here with a drug that enters the brain, impacts on some of the underlying biology and has some clinical effects. And those opportunities can be grouped in many different ways. They can be grouped in terms of the sort of fundamental research, understanding mechanisms, improving drugs and drug delivery, and as well as looking at non-amyloid targets, as well as galvanizing the much needed investment we need in both the basic science and the clinical research. From a clinical research perspective, what does this mean for clinical trials of Alzheimer's, and for that matter, non-Alzheimer's dementia? How can we look for trials to demonstrate maximum benefits, maybe going earlier, longer, comparative designs? We now have an opportunity to start to validate some of our existing biomarkers as well. And very importantly, what does this mean for clinical practice? Using this momentum to make what most of us will believe are long overdue changes to our services. We need to prepare for this drug or these drugs as and when they become available. But this means improving diagnosis and care now. And just as we've seen, say, for thrombolysis for stroke, there's huge amounts that we can do in improving care and outcomes just by galvanizing and improving our services. So the program that we have today is we're going to have a series of quite short talks. I'm going to pass over shortly to my colleague at UCL, Dr. Catherine Mummery, who's been involved in a lot of amyloid trials, who's going to give us an overview of the lecanemab results and touch a little on other anti-amyloid treatments. I will then pass back to Bart the Strooper for his views on the opportunities and challenges this means for fundamental research. My colleague in Cambridge and at DRI UK, James Rowe, to think about opportunities and challenges for translational research, and then to Professor Clare Mackay from Oxford to think about opportunities and challenges that we all have for clinical translation. We'd then like to open this up to questions from the audience, and we're keen to have a range of different questions, not just around the trial results, but future research directions and challenges for real-world implementation. Again, please use the Q&A box to leave your questions and to vote for questions that you would like to be discussed. So without further ado, I'm going to hand over to Kath Mummery to give us a more detailed overview of the lecanemab trial and its results. Thank you very much, Kath.
Thank you, John, for that terrific introduction. I'll just share my screen. Okay, I hope you can see those fine. So these are my disclosures, and as you've heard from John, this is to be the beginning, an opening discussion about the lecanemab results and what they mean. You'll hear some themes through these talks. There are things John has just alluded to that I'll go into in a little more detail. I have to say that the Clarity team had 90 minutes to talk about the lecanemab results at CTAD. I have 6. So bear with me. But what I want to do is try and put this in a little context initially, then talk through the results and give you some of my initial reflection. So this has been a long time coming, 20 years of looking at anti-amyloid therapies with promising signals, intermittently followed by almost inevitably until very recently disappointment. Active immunization was where we started, and the study was performed at Queen Square by Nick and Martin Rosser, amongst others, prematurely stopped because of what was then called meningoencephalitis, but turned out, we think, to be ARIA. Removed amyloid, but was stopped because of adverse events. Early monoclonal antibodies then fell by the wayside with negative, very long, very expensive trials. But then we started to see some signals. So the PRIME study in 2016, lecanemab, suggested a signal was present in terms of change, cognitive as well as functional, though it wasn't measured, it wasn't powered for cognitive. That drug then got accelerated approval June 2021. The controversy around that and the results continue. But then we had donanemab, which showed dramatic amyloid lowering in the phase 2 trial associated with other biomarker changes and some cognitive changes. The phase 3 for donanemab reads out in March next year. We look forward to that. Gantenerumab has been around for a very long time at increasing doses, and the high dose graduate 1 and graduate 2 read out at the CTAD conference 2 weeks ago and was negative. There are some points I'll come back to about that trial compared to lecanemab. It did show changes in amyloid lowering, but not as much as was expected or as much as others. And it was negative in terms of significance on all other changes. Lecanemab, in contrast, was positive, as you know, and is going to be the focus of the roundtable today. And I'll go into more detail in those results. So you've just heard about the lecanemab trial, the 18-month phase 3 study. John outlined this and what the monoclonal antibody targets. The population in this trial was relatively early, so the majority of patients were MCI with mild AD, as well as a smaller proportion of patients. They all had PATH confirmed. There were relatively liberal inclusion criteria, so individuals were allowed into the trial with more comorbidities perhaps than had happened in other trial protocols. The treatment, as you've heard, was every 2 weeks, so not for the faint-hearted, IVIs of 10 milligrams per kilogram. And the primary and secondary endpoints I'll go through further just now. So this is the baseline characteristics of the population that went into the trial. And the things that I want to highlight are that this was a more mild population than in some studies. So 2/3 of them had MCI, 1/3 had mild AD. And the MMSE was around 25.5 across the 2 groups. For gantenerumab, it was 23. And so this group was milder than in Gant and in some of the others. In addition, PET centiloid, so the standardized measure of amyloid on PET, was around 75. In gantenerumab, that was around 100. This again suggests you've got a slightly less heavy load of amyloid in the brain when you start the trial. The population was more diverse than in some trial populations, and comorbidities, as I've mentioned, were allowed to a greater extent than in other trials. So 2/3 of the US population had 2 or more comorbidities. Primary endpoints, so CDR sum of boxes, cognitive endpoint. This showed over 18 months a slowing of progression in the treatment group versus the placebo group by 27%. This equates to about 0.45 of a CDR sum of boxes point over that 18 months, which potentially equates to around 5 months slower decline than in the placebo group. In terms of secondary endpoints, amyloid PET showed a significant from early on reduction in fibrillar amyloid burden on PET scan. And so they started at 75 centiloids. By the time they got to 18 months, the average centiloid was 23. That's lower than the entry point for the study. So 68% of individuals that got to the end of the study would not have been allowed into the study had they started with that level of amyloid. So there was a significant reduction of amyloid in this population. The other secondary cognitive and functional endpoints, as you've heard, so ADAS-Cog14 and ADCOMS, both cognitive endpoints showed a progressive slowing by 26% and 24% respectively at 18 months. The functional endpoint, ADCS-ADL, which is a clinician I like, showed a slowing by 37% at 18 months. And as John mentioned, they looked at quality of life scores in addition. And one example is the Zarit burden, which measures carer burden over time. And they showed a 38% reduction in the worsening that individuals on treatment saw relative to those who were on placebo. Turning to the biomarkers. So biomarkers for tau showed a significant change both in CSF and plasma in terms of reduction in tau accumulation compared to the placebo group, which increased. The neurodegenerative markers were mixed, though. The total tau showed the same sort of change as we saw with phospho-tau, but neurofilament light did not show a significant difference from placebo to lecanemab in either the CSF or in the plasma. These are sub-studies, but there's large variability looking at the graph of CSF, NFL. And synaptic markers were encouraging in addition in terms of plasma and CSF. ARIA are always something that comes up for discussion quite rightly in these individuals. This is the most important AE in terms of potential impact. ARIA-E in this study were 12.6% of the population. That is significantly lower than a number of other trials. For example, gantenerumab was more like 48%. That is driven by the APOE4 homozygote population who have a much higher risk than the non-carriers at 32.6% of those had ARIA-E. The symptomatic ARIA-E were a small proportion, so 2.8% of individuals that were in the study had symptomatic ARIA-E. 92% of these, or 92% of all ARIA-E, were within the first 6 months. And the second most significant abnormal event was the infusion reaction, was slightly higher than most studies, 26%. And we need to perhaps consider why that is. As John mentioned, there was no excess mortality in the treated group versus the placebo group, and no ARIA-related deaths in the randomized controlled trial. So in summary, the drug met its primary and secondary endpoints versus placebo at 18 months. Biological markers suggested that there was sustained amyloid lowering, and downstream markers were suggesting disease modification. ARIA-E does occur. There is a lower incidence than some of the other monoclonal antibodies, but there is a higher risk in APOE4 population. And my initial reflections, I think firstly, this drug has clear evidence that lowering amyloid levels changes disease in a trial completed as per protocol. It's the first time we've seen that. There are consistent findings across a number of monoclonal antibody trials now that we're seeing significant reduction in amyloid. In addition to that, we are seeing downstream change, and we are seeing some cognitive signals. And I think what the evidence suggests, and this is borrowed from Professor Nitsch, who gave a talk at CTAD on the last day. If you look at the positive trials, lecanemab phase 3 and phase 2, and the donanemab trial, they all reduced amyloid below 25 centiloids. Engage Graduate 1 and 2 failed to reduce amyloid that much. And Bart may well be coming back to that later, as he has written a really lovely paper on this with Eric Karen. But it looks like the amount of amyloid depletion, the rate you do it within a trial, and how long you were at that low level matters. But there are, as John mentioned, questions. So what happens over a longer period of time? Is there a cumulative effect as the drug is continued, or does it stay at the same effect or even reduce? We may well find out from some of the trials that have now had people on drug for a number of years, albeit an open label extension. When should we be giving it? Is the trial population right, or should it be going earlier? And most importantly, when should we be stopping it? How do we manage comorbidities to minimize any additional impact or risk of these drugs? How do we predict both ARIA and outcome of ARIA and minimize those problems? And in the future trials, what are we going to do in terms of combination therapies? And one trial has just started, which is a combination of lecanemab and an anti-tau immunotherapy of the DIMTU population. The complexities of understanding that sort of trial are not to be underestimated. And most importantly, in some respects, how on earth are we going to implement these complex drugs that need very close safety monitoring in the UK, where our service really isn't fit for purpose at the moment from that perspective? And I'll stop there. I'm now going to hand over to Bart Strooper, the director of the DRI, who is going to really go into the opportunity of how we might enhance research and untangle some of the things that lecanemab and other monoclonal antibodies do. Thank you, Bart. I'll hand over to you now.
Thank you, Catherine. Fantastic summary. So my main question is basically, why did it take us so many years to come here? So it's a hypothesis which has been investigated for at least 20, if it's not closer to 25 years, with a lot of discussion. And I'm not going to repeat everything which has been said, but I think that it reflects mainly one big thing here in the field, and that's our lack of knowledge. So we have a huge lack of understanding how these antibodies work, for instance, but also a lot of other questions. And I think that the only thing we should do is now that we have a first positive result, is to enhance our investments and go faster, because we should have tested many of these things in parallel and not in consecutive fashion. So the big question, basically, I think for basic science is what are these antibodies doing? How do they work? And why have some antibodies which remove amyloid plaques no effect, and others have an effect? So how do we understand that? And here is a very nice scheme which Eric Aran developed. And he says that when you test an anti-amyloid antibody, the first thing you need is to remove amyloid to below a certain level. And he took the 20 centiloid as the lowest level. You need to remove the amyloid to a level where the brain can start to recover from the damage that is caused by amyloid. And so it's only then that you will see a deviation from the progression of the disease. And it's only then that you will see an effect. And so if you think about the clinical trial, and clinical trials are limited in time. So if your antibody takes more time to remove the amyloid than another, then that antibody which takes more time will take more time also before you go under the critical threshold, and the chance that you see a clinical benefit in your trial will be much lower. And I think that vibes with the learning that Kat just mentioned. But this is of course looking to it from a pharmacological clinical trialing perspective. But the big problem is basically, how do these antibodies work? And so what we know is that Alzheimer's disease is a very complex disorder, and in fact we are treating something which starts 20 to 30 years before we have the symptoms of Alzheimer's disease. And so there is a lot happening between the accumulation of amyloid and the changes in the amyloid biochemistry and the final result where the patient starts to live with dementia. And so we call that a cellular phase, and that's basically all the reactions which happen in the brain to keep the brain in homeostasis. And so the funny thing is that we use these drugs, that we develop drugs for this phase, and that we test them in fact in a late phase of the cellular phase, where we don't know very well what's going on. And so it's very likely that part of the cellular phase, which is the reaction of the glial cells, the vasculature, also the neurons, that part of that reaction becomes irreversible and cannot be reached anymore by the drug. So if we think then about treatments, I think that there are two reasons why the antibodies can only be the beginning of the story. The first reason is, of course, that they are incredibly expensive, and so one should wonder how we are going to make this drug available for everybody who needs it. And the second reason is that this is a treatment for this phase of the disease, and we need other treatments to treat patients which come in this phase of the disease, probably in combination with the amyloid therapy. So tau necroptosis, the glial reactions. TREM2 is one of the favorite targets there, but there are probably many more. And I think we should move away from this clinical phase where we have severe damage to the brain, either synaptic, which is maybe reversible, but also neurons which are dying. I don't think that, although we think that we want to treat there, I think that the real aim should be to target here. So the last point I want to make is about amyloid and the fact that these antibodies are very expensive. I think that, we know already for a long time that that genes in amyloid, mutations in amyloid precursor protein and penicillin are sufficient. It's all what you need to get Alzheimer's disease. And so we know also that, and Kat alluded at it, this whole story with the antibodies started with an accidental observation in mouse, where active immunization basically was able to clear all the amyloid plaques in this mouse, and later also in patients. Active immunization has been stopped because of the side effects, but I think that this experiment with lecanemab basically shows that we can generate antibodies which are protective. And so I think one way around the expensive drug that this will be is to restart the work on active immunization and think whether we can circumvent the side effects which have blocked further progress in that area. But on the other hand, since this lecanemab also proves that amyloid is at least part of the problem, we should also reactivate the thinking about the enzymes which generate amyloid. And as you know, many trials have been done on base-1 and gamma secretase, and they have also failed basically for side effects. But as we have argued in the past, these trials have quite some problems, and we can still improve a lot on these trials. And so I think that the lecanemab trial also teaches us that we can do that. And so I want just to show you that in the meantime, we have learned so much more about these two enzymes that I think the time is there to invest in that type of drug development again. And so here you see, for instance, a paper published in Cell last year from the group in China led by Jinhong Shi, which shows you cryo-EM structures from different gamma secretases with different inhibitors and different modulators, and which all have very different binding modes. And I think this opens really the possibility to make more safer and new drugs, but that's for and hopefully for the next seminar. And so this is the end. So thank you very much for your attention. And I'm pleased to pass the word to James Rowe.
Thank you, Bart. Let me just share my screen. Thank you. So, for some reason that triggered a quick cut out on Zoom. Can you see my screen now?
Yes. We can see your screen, yeah.
Fantastic. Thank you. Sorry about that little glitch. Yeah, so I'm going to actually just first of all just declare my declarations and disclosures here. I'll leave that on for a second, just for people to look through. The short summary is nothing of relevance to the material that we're talking about today. I'm going to begin actually with the quote from this review that encouraging progress has been made in the development of treatments that reduce disease activity, but the relationship between disability and such disease activity, as reflected in imaging, is complex, and evidence suggests it may be possible to delay the not yet abolish progression. And it's very telling. This comes from the great Ian MacDonald 30 years ago in relation to disease-modifying treatments for multiple sclerosis on a review of interferon. One of the challenges they faced as a community was that their imaging markers on MR were showing 90% or more reduction in those markers of disease activity. And yet the clinical effects, even on perhaps surrogate clinical events, and in their case, relapse rate, it was much more modest, perhaps 15 to 30%. And it was actually some years later that there was evidence of an impact on overall disability. And their community was wrestling with issues that we face too in the dementia world around natural history variability, the impact of study duration, and what can be expected in efficacy trials. But actually, the end of that review and the mood at the time was that results of those trials are grounds for optimism, that caution is still necessary, but progress now may be faster than before. And I think that's a fantastic sentiment to keep hold of and would apply today. So let's think about lecanemab, and I want to set it in the context of other agents. This is from Jeff Cummings's fantastic review from earlier this year, and hopefully there'll be an update early in 2023, reminding us that actually there are now 143 agents tested at the moment in nearly 200 clinical trials, 4 out of 5 of which are disease-modifying treatments, either small molecules or biologics, with some symptomatic trials ongoing. And the disease-modifying therapies, again, only 20 were targeting amyloid. You can see the numbers there targeting tau and inflammation and synaptic health. And I think it's really important we remind ourselves that amyloid is in the context of quite a complex pathophysiology, and indeed the effects of amyloid treatments may be conditioned on and partly mediated by tau and inflammation. So the hope is that we can take the very positive signals from Clarity AD, the lecanemab trial, forward. We don't lose interest, lose focus on the other possible ways to treat and prevent Alzheimer's disease, but we start to think about it in conjunction with amyloid-based therapies. And as Professor Mummery mentioned, there are indeed many different antibodies. She touched on lecanemab, aducanumab, donanemab, and gantenerumab that have shown these positive signals in recent months. And one of the interesting features is they seem to bind in a similar domain near the N-terminal of amyloid, in contrast to some of the earlier, seemingly less successful antibodies. So there's a whole variety of antibodies, and these differences may matter to their efficacy and indeed which patients benefit and their risks. But there are also many amyloids, and the antibodies have differential binding for the plaque that we tend to see under microscope, and which indeed is the main basis of the PET signals. And other antibodies, in fact, lecanemab is interesting here because of its preferential binding to the protofibrils that may indeed be more toxic than the plaques. Ongoing research needed to really pin that down, but I think the consensus is moving towards the idea of the toxicity of these oligomeric and protofibrillary forms. But even if that's resolved, there are some very important questions that come out of the lecanemab study and the other clinical trials. For example, is there a particular risk level of A-beta at which the risk of conversion to Alzheimer's or progression really starts to apply, or that may trigger secondary processes such as tau toxicity and this toxic alliance between A-beta and tau? Is there indeed a target level of A-beta below which one has to get following treatment in order to expect clinical benefit? And Professor Mummery touched on that, drawing attention to the difference between these more successful and less successful recent trials in that centiloid score achieved after treatment. And there's very interesting questions, in fact, which I encouraged through the lecanemab study, looking at the effects on tau and some of those inflammatory markers. And one can speculate that this alliance between amyloid, tau, and inflammation may need to be harnessed jointly in order to get best effects. And all of that in the context of what's really very dynamic disease and indeed dynamic treatments. So the accelerants of Alzheimer's disease and its progression don't have to be the same as the best targets for decelerating disease. There may be a difference there. And also the disease and the treatment effects may be non-linear. And I'll just touch on that with some cartoons on the following slide. If we think about the progression of disease over time through that preclinical stage through to mild cognitive impairment and on towards early dementia, there may be a non-linear severity seen here with this progressive acceleration of decline. If we think about how it might be preferable to treat at the earlier stage, and there may indeed be most to gain from treating people earlier as possible with Alzheimer's disease pathology. Actually, because the rate is slow, even if the drug treatment is effective, it becomes very difficult to show that. The signal to noise on the rate of change is difficult. Whereas if we think a little bit later on in early dementia or more moderate dementia stages, when actually there may be less to gain, arguably, missed opportunity for early treatment. Paradoxically, that rate of change may make it easier to demonstrate a change. And I think that's going to be very important as we look a little bit deeper into lecanemab results, where the aim was to treat early, as Catherine Mummery highlighted, but the response seemed to differ between those at MCI and AD if you dig into the supplementary data. That may not be a treatment effect, it may be more to do with the differences in power in a non-linear disease progression. We can also think around what effects we might expect to see on those time curves over the years of treatment. Now, if there was a symptomatic treatment or a disease-modifying treatment that became counteracted or compensated for by secondary disease processes, one might see an initial divergence of the cognitive decline slopes and then moving in parallel. So that's one option. Of course, I think what we'd all love to see is from very early on an initial slowing that was sustained, so an immediate and sustained disease-modifying effect. But because of the time taken to clear amyloid, and indeed that potential relationship between the speed of amyloid clearance and the risk of ARIA, it may take time to develop that therapeutic effect and only then to see the benefit through clinical measures such as cognitive scales. So one may have a lagged phase followed by this more favorable separation of the curves. And if that's the case, it puts a lot of pressure on the design of studies and the duration of a study, in that the longer that lag, the disproportionate the penalty is on the power to detect a drug effect after 2, 3, 4 years, any fixed length of trial. So it puts real emphasis on that trial design and investment. So where do we take all this? So first, I think we need to look to our regulatory colleagues to really dig deep into the weeds, to look at safety and efficacy, for example, to ensure that the actually really very thorough sensitivity analyses have done their job to a regulatory satisfaction to offset, for example, the potential for functional unblinding through ARIA. There were some really nice analyses provided in the paper, but I think we need to have consensus that they were sufficient. It's essential as we go forward that we engage patients and families. It's their voice really needs to be heard. We've heard a lot since the announcement of the results. Twitter is very alive with discussions of the risk and efficacy, talk of the minimal clinically important difference. But we need to think about who's bearing the risk, which is our patients, and who will see the benefits, our patients and their families. And that patient voice is going to be essential. And actually a comment that I've not heard echoed very much, but it's right there in the abstract and echoed in the main paper from the lecanemab study, that longer trials are warranted. This is not the trial to end all studies. It's the beginning of a longer journey, and longer trials are needed. And as part of those trials, we want to start to see a switch towards representative and what we might call real-world cohorts. Now, lecanemab study in Clarity AD did well in terms of comorbidity and some progress on previous trials in the diversity of their participants. But if we really want to think ahead about to whom might we want to apply such a treatment, how might we stratify or optimize treatment by stage or gene, there's lots of information within the lecanemab data to build hypotheses, but those will need to be tested and not just drawn retrospectively. So clinical trials evidence is going to stay important. Further work definitely needed, particularly I think around the MCID, the minimal clinically important difference. So this is part of a journey ahead, and that will build on and iterate with some of that mechanistic biology that we've heard from Bart Strooper in the previous presentation. So with those thoughts in mind, and with looking towards clinical trials and how we may apply treatments such as lecanemab, I'm going to hand you over to my colleague, Professor Clare Mackay in Oxford. Claire.
Thank you very much, James. Okay, thank you very much, James, and to everybody, John, Giovanna, etc., for an opportunity to talk a little bit about how on earth we're going to start thinking about scaling our capabilities in the UK so that we can trial first and ultimately deliver these sorts of novel therapies at scale. My disclosures are short and are down in the corner there, and while I just leave those on for a moment, I'll just say that as we're These early trial results are so exciting, but as soon as we go beyond this, it's a phase 3, but it's still very early days. Whatever we do next, however this gets regulated, whether it gets regulated, whether it gets licensed, etc., we need to be thinking about our capabilities in relation to trials and delivery. This is the screening and inclusion table from the Clarity AD trial. You're not supposed to be able to see the details in there, but I just wanted to use this to highlight what we're up against. So in order to get access to this trial, you need to be diagnosed early. So early diagnosis, so MCI, as Kath said, is 2/3 of the population, and early AD making the other third. So being able to access patients early is already a significant challenge. To get into the trial, you needed to have an amyloid status measured. That's by either PET or CSF. There then needs to be a 2-weekly administration, intravenous administration, infusion of the drug, and then followed up with regular MRIs to check for the safety, so the ARIA contraindication to the use of these drugs. And then, of course, if we're thinking about the trials, you need all of that same infrastructure to be able to get the endpoints from CSF, plasma, and PET alongside the easier to access clinical and cognitive scores. And if you think about this in contrast to what most people have available in terms of the assessments within a memory clinic setting at the moment, there is a mountain to climb. Around the UK are a group of individuals who are starting to think about what we might need to do with services in order to drive our interventions earlier. We're calling this the sort of nascent brain health clinic network, and there are a number of different models, a number of different services, and a few of us who are sort of putting together the first thoughts about best practice in relation to a number of these things. But the key drivers are largely in common, and they include going earlier, having high quality biomarker assessments incorporated within clinical studies and clinical practice, an emphasis on personalized risk reduction, risk management, and the alignment of service and research so that we can be accessing large-scale real-world populations and to be able to precision recruit for the right patient to the right trial at the right time. I'm just going to spend a slide describing the pilot that we've run in Oxford in a psychiatry-based memory clinic, and this is, I think, fairly typical of psychiatry-based memory clinics around the country. There's a lot of variety, but nevertheless, in our service, it might have the word Oxford in it, but it's a very typical service in that GPs refer into the service, a triage happens, and a decision made about whether a scan happens. It's virtually impossible to get anything other than a CT scan for all of these patients. The patient then has the CT scan en route to their memory clinic appointment, where that is the one time when the patients will see a doctor, where they're either diagnosed and discharged or potentially followed up if they're earlier stage or there are other reasons. So what we did in Oxford was to divert people away from the CT scan that they would have had and offer appointments in our brain health clinic, where we offer high quality assessments as standard and research assessments as extras for everybody coming through. Everybody coming through the door is asked if they are interested in taking part in research, and they can say no, and that's fine. And if they say yes, we give them options. So they can say yes to the use of the data that we're collecting anyway. They can say yes to having additional assessments while they're in the building, and they can say yes to recontact for future studies and trials. And we put together a protocol. Our sort of speciality in Oxford is around the MR imaging. So we've aligned our MR imaging with the UK Biobank. Other than that, there's nothing particularly unusual about what we're doing, the cognitive and clinical assessments, etc. We have ambitions to incorporate more blood-based and fluid biomarkers in the future. And the reason for explaining this to you is because I think if you put together the infrastructure like this, you can create this sort of win-win-win between patients who are getting a thorough assessment and the potential for earlier and more accurate diagnosis, and the potential for stratification into the right trial for them. We have been running this pilot for about 1.5 years, and we've got excellent feedback from all the patients that have been through. The clinical service loves it. We've actually been able to reduce clinic appointment times with the way that we've set up the service for them. We've done a qualitative study in which clinical staff express surprise, happy surprise that patients are able to tolerate the sort of additional assessments that we are providing for them. And then on the research side, we get above 90, 80 and 70% consent in those three categories that I described. So a really highly representative research cohort that is developed on the back of this, with the potential for precision recruitment for studies and trials. I think that the only way that we can make this work is to align all of the infrastructure that we have at our disposal. And so I'm thinking that, for example, this sort of growing brain health clinic network ideally bolts straight onto the joint dementia research infrastructure, which would make the individuals who've consented for recontact for future studies and trials available to the largest number of studies that are out there. We want to align, obviously, with the Dementias Platform UK. We're part of the Dementias Platform UK, where both the patients who are coming through, who are entering services, can be put together with patients, with participants who are part of large cohort studies, so that we can identify those very early patients who are going to benefit most from the sorts of interventions that we've been hearing about. Another thing that I just wanted to finish by highlighting from this slide is the screen failure rate. So as you can see, there were nearly 6,000 people that were screened for the Clarity trial, and 70% of them were screen failures. And so this really just highlights what many of my colleagues have already said about this being a really exciting time with the first potentially disease-modifying drug starting to look promising, but there are a lot of patients for whom other alternative interventions are going to be needed. So that's really my take-home message. It's really exciting times, but there are currently very few places in the UK that could administer a drug like lecanemab, and very few people with the skills to interpret the sorts of data that we're talking about. So we need a massive effort in upscaling our infrastructure, a massive effort in training an army of people who can administer drugs like this and contribute to trials like this. We urgently need this service innovation. The brain health clinic models are a start. We can have a look across the models that are existing and start to see how we can develop infrastructure so that we can reach the maximum number of people, and particularly reach those people who are further away from the sort of real centers of excellence that we all represent around this table today. And we should, I think, be aiming to align the existing infrastructure and to create this win-win-win for patients, clinical service, industry, and government. So as all of my colleagues have said, this is just the start, and there's plenty for us to get our teeth into. So I'm going to hand back over to John, I think, and we start the questions now.
Thank you very much indeed, Claire, and thank you everybody, and thank you everyone who's contributing to the questions that are coming in. We're already running short of time, so we're going to have to be quite quick about this. Can we start off with one just quickly for Claire, just about what's the funnel into the brain health services? Can people refer themselves? Do they come via GPs? What's the route in?
So our pilot was all about integrating with the existing service. So it was GP referrals into the secondary care system that were being diverted into the brain health clinic. So we have essentially been targeting our efforts into to those who are at the point of entry into secondary care, which is in many ways the easiest place to get access to people who are in the current system. Of course, we, like everybody else, think that earlier is better. But that's a whole different challenge of how you develop the infrastructure to enable people to self-refer or get GPs to lower the bar to referring in. And obviously that creates a huge potential resource requirement. So our pilot was all about integrating with the existing service.
Thank you. So we got a question around the potential sex differences which was shown in the supplementary. Kath, would you like to take that?
No, absolutely. This is an excellent question. as a woman, we should be looking at sex differences better than we look at sex differences at the moment. So thank you for raising this. I have had the opportunity to have a quick look while we've been talking at the article that you quoted in your question. And she does a very good job of defining the pros and cons of taking subgroups in a forest analysis. I think for this particular trial, there will be follow-up papers that will look at, I've been told, will look at those subgroup analyses in much more detail. So I don't think, and I hope it won't be brushed under the table, there will be explanations of what people think is going on. I also think we have to be careful, as she's written in her article, these are forest plots. And you have to be cautious when looking at subgroups in forest plots, especially when the confidence intervals overlap. They all do go in the right direction. So she's mentioned the CDR-Sum box's primary endpoint. When you look at the other cognitive markers, ADAS-Cog, and you look at the functional markers, in all of them, you see a benefit in both male and female, but you see a greater benefit in male than you see in female. So in female, it's more 15% to 30%. In male, it's more 35% to 50%. So, but with overlapping confidence intervals. So in short, I think we have to be careful about how we look at subgroup analysis and make conclusions from them. It looks like things are going in the right direction in both groups, but I completely agree that we need to understand the model changes much better than we do at the moment. And I think, John, you wanted to add something to that.
Yeah, I think it plays into our rather lack of understanding about why women can get more Alzheimer's disease generally, and a lot more research in that area. Absolutely. So I think the next one we'll go to Bart. It's a very interesting question from somebody who I know probably has an angle on this question, which is that the antibodies here were raised against protofibrils from people with the Arctic APP mutation. So does that mean that we can now say that familial Alzheimer's disease and sporadic Alzheimer's disease are part of the same disease? And my supplementary question would be, is there something about targeting the protofibrils? Your work has shown that it's likely to be towards the plaques and the protofibrils rather than the soluble forms. Is there something about targeting the protofibrils that we think is particularly beneficial?
Yeah, well, I think I will start with the second question. So obviously, if you have the monomers as target, you will drain a lot of the active component just away by binding it to this monomer. So there is obviously an advantage to try to target more pathological conformations. I'm not going to go into discussion about oligomers and amyloid plaques because that would take us too far. So then the other side is about it's for me a very strange question, because I have never hesitated that familial Alzheimer's disease and sporadic Alzheimer's disease, that it's a large spectrum of a very similar problem, and that amyloid is an important component of that. And I think that the lecanemab has finally shown that this is true as a claim. I'm also very far from very simplistic ideas that it's all straightforward and all the same. I mean, there is nothing more different than people, and there is even there is nothing more different than the brains of people. And so, I mean, these are all, I mean, there are some fundamentals which are shared, and I think that the amyloid is there, is the amyloid peptide is that. And then there are a lot of differences. Everybody reacts differently to this thing. So shortly, I think that this was never a serious difference, and I think that this trial basically proves that that the essence is very similar.
Thank you. We're going to move an interesting question which relates to potential differential effects of what we measure as tau, so of phosphorylated tau in CSF and blood, and then what we might see in tau on the tau PET scans. James, I don't know whether you are in a position to comment on that.
Yes, but I might begin with the PET question and then come back to the CSF. The PET is very valuable for quantifying and localizing both amyloid and tau in the brain. But we have to remember that for amyloid, the current PET ligands largely have affinity for plaque amyloid, not fibrillary amyloid, so not those that might be most toxic and not those that are targeted by lecanemab. So that's an important disconnect. Similarly, for tau, the current generation of tau ligands bind to mainly tangled tau and not to toxic oligomeric species. So it's a useful indicator, perhaps for global tau burden, perhaps even after those relatively protracted processes of aggregation. but it may not be a simple surrogate for engagement at the molecular level, target engagement onto either oligomeric tau or protofibrils for amyloid. And that's a really important difference. So I think would be helpful, and we would, I think, seek comfort from it there being a reduction both in the amyloid level and in the secondary tau burden, but it shouldn't actually be that sort of killer question. There are reasons why drugs may work without changing PET signals, and indeed you can change the PET signal perhaps without having a clinical effect. So this is useful adjunctive information, but it comes back to the question, there are many amyloids, there are different forms of tau, they have different properties and different significances for the disease. That similarly applies when we come to measure them in CSF and blood. There's a question around the assays, different assays for phospho-taus, whether it's 181 or total phospho-tau 181, 217, 231 may differ. But that aside, they may have differential properties for predicting progression at different stages of disease. That's ongoing work. But it is useful. So the reason the blood, I think, is especially useful is the scalability. One can do frequent sampling, frequent monitoring much more readily than one can for PET and even some of the more intensive, inexpensive MR and cognitive assays. So we're trying to balance scalability and frequency of sampling with the proximity to that clinical syndrome, which is our ultimate goal. All of these measures, I think, are best used in conjunction and playing to the strengths of each assay, but being aware of their individual weaknesses.
But it's also fair to say that there is some tau PET data on lecanemab, which does show.
That's right. So there's quite a major sub-study of some hundreds of participants who came into having tau PET. I think the full data on that is yet to be released, but there are some encouraging signals that have been presented, at least at a conference level, of reductions in tau PET. It would be interesting when we get those data to actually understand this interaction between amyloid and amyloid therapies and tau. Is tau merely going to help stratify who benefits from anti-amyloid treatment, for example, or maybe the secondary effects on the tau burden will be an indicator of the mechanism of benefit? So there's lots to be done here. And I think, as Bart said, this is not a simplistic model of amyloid or a singular amyloid for a singular disease. It's going to be much more complex, but we have the tools to pick apart the story now.
I think we've got some other questions on biomarkers. We might just go to different topics to just keep things moving a little bit. So Claire, I'll come to you, which is probably one of the knotty questions. In fact, we'll get, I think, probably lots of people's views on this, which is a treatment of 0.45 on the CDR sum of boxes, is that meaningful? And in terms of service development. So Claire, would you like to start with that? You're muted, Claire, sorry.
Strategic muting, because I just like everyone else don't know the answer to that question. I think that in the end that question is about what patient, in many ways the answer to that comes down to what patients find meaningful to them, and there are various ways in which we can try to measure that. I'm going to try and bounce that one back to one of my clinical colleagues who probably has more of a feel for what would be meaningful in the context of having a patient in front of them in the clinic.
So from my perspective, and then we'll go briefly to Kath and to James as people seeing patients in the clinic, is that one, there's clearly there's huge variability, and defining those sorts of things on various scales is another way of just trying to get a handle on what is significant, often using statistical methods. What strikes me is that there are data on quality of life in this study, and in the UQD we've got improvements, and in quality of life measures we've got differences, and interestingly we've got measures on the Zarit burden for carers. So I think it's important that we look at these all together rather than just in isolation, and choosing a number on the CDR sum of boxes is arguably, I would argue, is perhaps less relevant than looking at some of the outcome measures that we have, which are more directly measuring what is of interest. Kath, James, would you like to come in on that?
Sure.
Catherine, go first, yeah.
Thanks. So John, I completely agree with you. I think we need to look at everything in the round. Obviously, CDR sum of boxes is the primary endpoint, so we do need to consider that very carefully. It was significant. Is 0.45 clinically meaningful? If you have somebody in front of you and you can tell them that they're going to take 5 months longer to get to a particular stage, they might find that clinically meaningful, because they might still be able to say hello or goodbye to their grandma, recognize their daughter, whatever, for 5 months longer. That's potentially, it's small, but it's meaningful. I think we have to remember that these are potentially disease-modifying therapies, and what happens over time. 18 months is a very short time. And some of the data that's coming out post-hoc on patients that have been on drugs for longer is suggesting that the effect is cumulative and it increases. We need to see a lot more on that. But I think that that's critical, monitoring further if lecanemab goes through to see what happens over time and how much those two diverge. And I agree with John that functional scores are so important to patients. They understand if they can cut something up when they couldn't, they can't cut it up the week after. They know what that means, whereas CDR 0.45 or CDR 0.5 means nothing to them. One last point, which is perhaps slightly flippant, I don't mean it to be, but in terms of the scale and 0.45 versus 18, well, the Fahrenheit score is 212 degrees, and 3 on there is meaningful for most human beings. So I think, you know, there are ways to look at this.
James, do you want to come in briefly?
Yes, I think there's much discussion of this and whether the curves of the serial assessments are parallel or diverging, and what that means. We have to remember that although when we look at those curves, to my eye, they appear to continue to diverge over serial assessments, the study was not designed and not powered to look for that through interval assessments. It was for that very specific, difference at the end point. And we shouldn't sort of do retrospectively our own sort of mental gymnastics, applying retrospective statistics to tables and figures which are just not designed for that. I think it is encouraging, not just that I think that, despite what I said, that the curves appear to diverge. More importantly for me is we're getting consistent effects in primary endpoints on cognition and secondary endpoints, cognition, function, quality of life for the carer, at least, the desired burden. All have actually very similar effect sizes, 25 to 50% slowing. And I think that a 25 to 50% slowing of the disease and expression of disease and symptoms, I think that will be seen to be worth having. Ultimately will not be my decision. We need to be engaging carers and families in a discussion with regulators. There's a lot of discussion that we have to be looking for MCID of 1. That comes from anchor-based studies, which is one method to determine MCID, but it shouldn't be seen as the only method to determine a minimal clinical change. So I think it's extremely encouraging, but the bigger the effect, the better. But I think this effect we have is enough to take interest and to change our thinking as we go forward.
And in answer to a very specific question in the chat, which is around the EQ5D, that was measured, and I think there was a 44% decline in the treated group. Perhaps we'll come back to you, Bart. The question, I guess, is there's a question really around not just focusing on amyloid, but thinking about other targets. And I guess there's a question about what are the other brain targets that you think we should be particularly focusing on?
Well, I mentioned them briefly in my conversation, the glial, astroglia, microglia reaction, where there is quite some genetic load is certainly potential area. And then tau is the obvious idea already for decades. The only remark I want to say is that we are very far, if you compare it to amyloid pathway, we know so much less about those pathways. And I think simply extrapolating that an antibody against tau will work because the antibody against A beta will work, I don't think that we have any ground to assume that. Said that, it boils all down to investment. For COVID, in 4 years, we invested more money, or at least there are more publications on COVID in 4 years than in 100 years of Alzheimer research. So if you ask me these questions, then I have to say, give us the means to get there. So
lots of new targets, just much more investment needed, and going and really understanding what we're dealing with. Yes. Which I think leads us nicely to a question from Martin Rosser, which is around these sort of differential neurodegeneration markers. And for those of you who don't know the data so well, we're showing really quite consistent changes on phospho-tau. We're showing some changes in tau within the medial temporal lobe. We're showing some changes in GFAP and astroglial marker. We're showing some changes on a synaptic marker in the CSF. We're not showing changes in neurofilament light. We're showing some hippocampal volume probable preservation, but probably some excess loss of whole brain volume, cortical tissue, and increases in ventricular size. So James, that's a really simple selection of biomarkers for you to unpick and tell us what's going on.
Yeah, thank you. An easy question, John. Thank you. I think we should remember that synaptic loss occurs before neuronal loss. You know, if cells do synapses. Synapse is the unit of information exchange in the brain. It's how nerves talk to each other. And indeed, in the neuropathology studies, synaptic loss is the closest correlate to the cognitive deficit at the end of life, even more than tau and even more than amyloid. So synapses, I think, are critical to the function of the brain. We now have a range of potential ways to measure synaptic, sort of aggregated or pooled synaptic health. You mentioned neurogranin, SNAP25, pentraxins, a range of different fluidic biomarkers. We also now have PET ligands that can measure synaptic density, which weren't part of this study, but are now increasingly in research use. And things like UCBJ that are very stable, well-behaved ligands. And we know from other diseases and increasingly in Alzheimer's how they also relate to disease severity and progression, not yet in the trials context. So I think this could be a real strong card for improving signals of efficacy related to function. I might also just mention that when we think about nerve function, there are electrophysiological methods like EEG and MEG, and there's some very powerful tools now coming through that can detect functional changes of the synapse and those changes over time with disease progression that we could also look to build into early phase or experimental medicine studies. They're not going to replace clinical efficacy as our ultimate goal, but they could certainly help support the selection of drugs or drug combinations that we would wish to invest in more.
Thank you. But I think it's fair to say that the mystery of the increasing ventricular size and the cortical thinning, which has been observed, Nick Fox showed this back in the very first phases of active immunization, still remains unclear and the subject of a lot of debate. In this study, the neurofilament light changes have been potentially put down to the time course, but some questions whether they might relate to ARIA. But the neurogranin changes are quite encouraging, as you say, that we are engaging with the synapses. So we've got a question which I think probably two together, which is around both patient population and the timing of the disease onset, early rather than late, and whether we think that might affect how much of efficacy that we see. So I guess, Kath, what's your thoughts on the timing of when we should be giving these medications to people?
You know me, John, I always want to go earlier, but the tension there is running a trial earlier is longer, more expensive, and harder to show outcomes in, which is the problem with the decisions with all of these trials, is that there is that tension. They want to see people progress, but at the same time, to progress, as James showed earlier, you need to go later. So I think that is really tough. I think we should be giving these drugs earlier than we're giving them in the clinical trials at the moment. Ideally, we just need to have better markers of proxy. I think that the difference in many ways between, as I mentioned it a few times in my presentation, in many ways the difference between gantenerumab and lecanemab was that the gantenerumab population were enriched for recall issues, which effectively made them later. Their centiloid values were 100, not 75. They had much more amyloid in the brain, which means at the same time scan, even if it was as quick as lecanemab, which it wasn't, you wouldn't get it down as far. And I think, as Bart has shown beautifully, that really does seem to matter. So if you go early, you get a better chance of being quicker to get the amyloid down, decreasing toxicity, and perhaps even getting to stability.
Great. Bart, perhaps a question for you about getting these drugs across the blood-brain barrier.
Yeah.
What are the barriers, because it's an obvious barrier, but what are the implications for that, for this drug and for future trials, future therapies?
That's really one of the things I've never understood in the development of these antibodies, because it's really a very small fraction which gets into the brain. That's also why it's so important not to get a peripheral sink where all the antibodies are held. So it's a very low percentage. So it's really a waste of a lot of money. I expect a lot of modifications of these antibodies with blood-brain transport transporters, which even if you increase it only from, let's say, 0.8% to 4 or 5%, that would be a tremendous profit because they are expensive drugs. So that would be a very good thing. So that's what I expect will happen. So there is really a lot of work to be done.
And there's some potential for using transferrin receptors and things to try and get drugs more efficient into
exactly. You can modify these antibodies, and there are actually experimentally already examples of that. So you can modify these antibodies to make them better transported over the brain. But I also think that we need to think about other approaches. There's also a lot of questions about cost benefit. Well, I'm not going to address that as for the clinicians, but I think that we need to think about small compounds. This is very similar to prevention of atherosclerosis, where you have a statin. I agree with Pat, we need to treat early and with a drug which is available for a large group in the society.
Kath?
Just to follow up to Bart, actually. I mean, firstly, completely agree that small molecule is really important. And the gantenerumab is going to be called something new. The shuttle version is actually still under study, which has an active shuttle to get past the blood-brain barrier. So my question to you is, given we've got 1% going into the CNS, unless there is a blood-brain barrier disruption, which we're having in ARIA, what is happening at that point in time, and how much variability do you think? And we're being flooded with drug when we have a problem with the blood-brain barrier. Have you got thoughts about that?
I think it's a fantastic remark, Catherine. I don't have any good guess there. I don't know whether this improves the transport even. That's for me unclear. And so there we also coming to the question whether these antibodies doing it. It's such a small amount which goes over the blood-brain barrier. I think they are more working like a catalyst than something which is cleaning up all these antibodies. And so this brings us to this idea about a glial, astroglial, microglia reaction. So we know that the Fc fragment of these antibodies is very important. So probably what we are doing, well, probably I'm not going to speculate, but there are certainly links to the cellular phase of the disease, and we need to understand that much better. So blood-brain barrier, the cellular composition of the brain, the genetic composition of the patients is also something which we will have to investigate in the future.
Thank you. So we're really shuttling around lots of different areas of discussion here. So a question that's come up a great deal, which again is I seem to go to James for the really knotty ones, is around thoughts about cost effectiveness and cost benefit, and what you think about that and how we might begin to approach that.
I think it's a really important question. Of course, the answer's in the question. We don't know about the cost yet, and we don't know fully about the long-term benefits. The cost, we don't know the cost of what the drug might be. That's very much will be up for discussion. We could have some guess as to the cost of an infusion service at scale and MR monitoring, and that's not going to be trivial, although there are already moves perhaps to try and deliver this in an easier subcutaneous fashion and other modalities. In terms of efficacy, I think a lot will hinge on whether the longer term studies seem to suggest a progressive benefit or not. One thing we do know, though, we do know the cost of untreated dementia. We know the cost, the human cost and the financial cost of not treating Alzheimer's disease. The UK alone, the figures from 3 or 4 years ago were 30 billion. That's almost certainly gone up now. Be 35, 40 billion a year on year. And the biggest cause of death before COVID, during COVID, and after COVID is dementia. So we know a lot about the cost of not treating dementia. I think that we'll look to our regulators, and then we're in the UK, we'll look to NICE in other countries. There'll be insurance agencies to be involved in this. So it is going to be important. But cost and benefit, we need to know before we can work out that magic ratio and make a decision about availability on healthcare.
Absolutely. Kath, you've got a comment.
Yeah, I mean, to James's point about not treating and the expense of that, absolutely, and it's not modelled properly right now. And it has been ARUK looked at it actually and did a really nice paper on it, but we need to understand that better. There was just an interesting aside from the clarity point of view. There was some modelling done, and it's modelling on the phase 2 data, not on the phase 3. They're going to update it with the phase 3, where they looked at length of time in milder forms, stages of Alzheimer's, and they showed that there was a longer time spent in milder forms of Alzheimer's disease, shorter time spent in later stage, and importantly, a significant reduction in length of institutionalization. And I think that sort of modeling and exercise is valuable in giving you an idea of what you really are doing over the long term. Clearly it's modeled, but it's still worth looking at.
There's a question that slightly troubles me with this, which is the length of treatment, because what we don't know is whether one's going to need to continue infusions every 2 weeks forever, which would seem impossible. But given that by the end of this study, 2/3 of the people wouldn't have been eligible to enter the study, whether there is work to be done in perhaps using blood phosphatide or other metrics to try and get people down to an undetectable level. And if we take the model that Bart and Karen have proposed of the sort of, I'm not quite sure whether it's the hit and run, but whether you need to get the levels down and then sit and wait, whether actually, if you look at that pronounced lowering of amyloid burden on PET over 6 months, whether actually that might be sufficient, because clearly treating people vigorously for 6 months would be better than 18 months. Doing it monthly rather than 2 weekly would be better, and doing it subcut and ideally orally would be better than giving it intravenously. Kath and James, I don't know if you want to come in on that. Kath?
Hugely important question. We've only had one trial that's shown an induction process and then stop and monitor, and that was lecanemab, which was beautifully designed. Every other study has continuously modelled, and I thought from some of the initial data we saw from various studies, including lecanemab, that the lack of, and donanemab, the lack of increase over time was encouraging, that we would have a period of just monitoring, like you say, with tau or similar. I was interested to hear the opposite opinion from, perhaps unsurprisingly, from somebody from lecanemab, who suggested that it wasn't kept down long enough. In order to reduce the toxicity, you needed to continually remove that amyloid. I didn't see the data for the basis of that. It was an opinion only, but there are differing opinions. We need more information. And I guess we'll get some of that from these studies where there has been a gap in terms of where people were on treatment, came off treatment, went back on it, and looking at the measures that we have in those. And Bart will be reading out next year, which is one which will hopefully show us some of that.
James, have you got anything to add to that?
Yeah, well, just to say, so that discussion is largely focused on the dosing regime. Do we spread out those infusions of the amyloid antibody to a maintenance dose, or wait and see and treat those whose levels of amyloid appear to rise? There's another aspect of this, which would be to get induction and control of the illness, for example, through an anti-amyloid approach, and then to perhaps rely on more maintenance with another target, perhaps an anti-tau. Once you get the amyloid down, you might then want to treat more actively the secondary tau toxicity. And there are studies beginning. I think the dominant inherited Alzheimer's disease network trials unit is beginning a trial of anti-amyloid and anti-tau treatment in combination. That's the first of those sort of combined trials that I think really that have been embarked on. So it's not just around the dose regime of, say, lecanemab. It would also be around what adjuvant therapy we might imagine adding in for longer term maintenance. It would. I'll leave it there.
Thanks. So Claire, we'll come to you. Carolyn Patterson, immensely experienced, has just written the problems of delivering this form of treatment, ? And other people are saying, perhaps more specifically, who are the population that we should be targeting if we had this treatment in our routine clinical practice?
Yeah, I mean, as per my previous answer around this, I think this is actually a really critical question, and who will benefit most is something that we ask ourselves a lot in relation to the brain health clinic that we have put together. One thing that occurs to me in relation to something that Kath was talking about earlier is earlier feels biologically like the obvious place to go to, but earlier people aren't ill. And so, you know, having to come into a clinic and have an infusion every fortnight for something when they're not experiencing any symptoms or very mild symptoms, that's quite a big challenge that we are. I think we'll struggle to get people to engage at that point unless they have been identified to have very clear, strong risk factors, familiar risk factors, you know, genetic, etc. But nevertheless, I think that convincing people who are not ill to come into a clinic and have an infusion every couple of weeks is going to be a bit of a challenge. I think that the other thing that I wanted to say in relation to this, and I noticed an earlier comment about how in neurology-based services, the idea of doing regular infusions is already in place for other indications. But the vast majority of patients with dementia in the UK at the moment are not seen in neurology-based services. They're seen in psychiatry-based services, gerontology, primary care, etc. And the availability of the sorts of assessments and the sorts of infrastructure you need to deliver that, if we think it's bad in neurology, where, you know, probably one of the biggest problems is the waiting list, outside of neurology-based services, those things are virtually non-existent. And so with it, you know, that feels like a bit of a cliff edge to me. And the shortfall of the people who are appropriately trained to deliver these sorts of therapies, I think, is absolutely massive problem and something that we need to start thinking about right now, or else, you know, how on earth are we ever going to be able to deliver this?
So there's problems around personnel, there's problems around infrastructure, there's some things that we may be able to get around. So some of the trials that are screening with blood phospho-tau may well make things easier. We're still going to need to work out how we regularly use MRI scanning to control for ARIA, of course. And I guess there are some thoughts in terms of the pre-symptomatic prevention. There are, if we can predict, again, more investment, so more research to predict who is at higher risk, we can follow models from familial Alzheimer's disease. And of course, there are studies like the AHEAD study, which are actually already trialing these and related antibodies in pre-symptomatic people. I was always struck by research from Alzheimer's Research UK that as the majority of people in this country would like to know whether they're at risk of Alzheimer's disease. Whether that plays out when that's a possibility, of course, like we saw in Huntington's, is less clear. But I think there is an appetite, and this is such a feared consequence of aging that that may well be a possibility. But again, we do want to make the infrastructure and what people need to go through a little easier. Bart, perhaps I can come to you, perhaps with what will be one of our final questions as we're running out of time, and apologies to those whose questions we haven't got to, which is around whether ARIA is good or bad and its relationship to ApoE4. Again, sorry if we're throwing you under the bus here, but it's an interesting and difficult question.
I think that is one of the things where people most speculate about. So there is certainly an increase in ApoE4 carriers. We do not really understand how it's induced. It seems to be related with inflammation. There's quite some speculation there. We discussed already whether it affects blood-brain barrier or not. I don't think we have a clear view on that. So I don't know. Maybe I don't know what I have to think about it. It's something which came up with the antibody treatments very specifically. So. Probably any of you have better ideas here.
There's also some interesting data about the ApoE4 homozygotes getting more ARIA, but perhaps getting less benefits as well. Maybe being a little bit more advanced in this study, which is an interesting question as well.
I can say that the critical thing is in that all-important outcome, the slowing of disease, the preservation of cognition. We can look to the data now when it's been fully available around the impact of REE and REH and its relevance for your long-term outlook, and whether there's a relationship between the amount of amyloid reduction and the cognitive slowing. These can be really important questions. So for all the speculation on mechanisms, the proof will be in the pudding of clinical benefits, which we care about for all our patients and families.
But it's also, again, looking to wise heads, Martin Ross's comment in the chat that we also, of course, need to be careful about not over-treatment and problems that can be done with diagnosing people with prostate cancer based on PSA measures. And that also leads to another question about people with MCI and who goes on to develop Alzheimer's disease. And of course, the figure probably is around 50% at 5 years. And we are getting some algorithms to better detect who those individuals are. But clearly, before we go to the pre-symptomatic phase, we need to get better at determining who comes to our clinics, who is going to progress and when. And my colleague Nick Fox has often talked about the need for a proximity marker. We're getting quite good at detecting the pathologies, but not necessarily when we're going to run into problems. I think having asked you a difficult question, Bart, I'm going to close up by asking one that I know will be right up your street, which was a question about should we be doing more around the gamma secretase inhibitors?
Of course. Of course, I mean, I'm not going to talk 10 minutes about this, but I just showed these cryo-EM structures of the gamma secretase. I mean, well, let's summarize. So, the gamma secretase had the unfortunate honor to be the first and was very rapidly left on the side because the pharma thought that they had better stuff. But I think that if you read the papers again, and I've published on that, if you read the trial, the main trial there was really optimizing for side effects and minimizing for effectiveness. But I can talk a lot about that, but that's maybe not the place here. But obviously, we should look at it again.
Great. So I think, unfortunately, our time is rapidly running out. So first of all, I'd like to thank my panelists for really, really interesting presentations and discussions, to thank Giovanna, Emilia, Liz and colleagues for putting this symposium together and for advertising it so widely. For the over 350 people who tuned in, which is really a testament to how exciting and interesting. If we said 10 years ago we get 350 people turning up to almost any dementia seminar, you would have been laughed out of town. So that's fabulous in its own right. And thank you very much for everybody's questions. I think we've deliberately tried to keep this broad ranging. Clearly, any one of the topics that we've discussed here, from the basic science to the translation to the clinical applications, could do with many hours and will do in the future. And I think on behalf of the UK DRI, we're very keen to remain part of this conversation, to be working with the other organisations in the UK and overseas, and the charities. And this, we hope, will be the first of many, many conversations. And just taking a step back to say what an exciting moment it is for our field to have treatments that clearly impact on the biology, have some cognitive and quality of life benefits. And so thanks very much indeed for tuning in, and we look forward to continuing our discussions. Thank you very much.
Thank you.
Thank you.
Thank you, John.
Bye.